Okay, so hello everyone, I'm Helen, and I'm going to talk to you today about how the Mexican cavefish is helping to mend broken hearts. Not talking about if the Mexican fish are in love and then they fall out. So who am I? So this is me. Talking about how I got here, my pronouns are she and her, and so for undergrad I came here to Oxford and I studied biomedical sciences and I loved it, and then I thought, oh, I don't know what to do next. Don't really want to get a job yet, so I'm going to do a master's, and as part of my master's I got to go to Australia to Brisbane, which is this lovely sunny city, which was a really nice break from the UK, and I did an awful lot of things like taking cute selfies with animals, lying on the beach and having a really nice time. I did also do some really cool science, where I worked in a cardiac regeneration lab, and that's how I sort of fell into this field. I'm now doing this PhD, and now I can't imagine ever doing anything else, but I really feel like I fell into science, so I fell into science, but being gay, obviously I didn't fall into. And I definitely identify with it. That's just me. And the reason I wanted to do this talk is obviously there aren't that many women in STEM role models, and I identify as the B within LGBTQ, and I feel like bi-visibility is a problem across, not just in science, not just in STEM, across the whole, films, TV, books, everything. Being bisexual, bisexual women exist, and we can also do some really cool science, which is what I'm going to talk to you about today. So my whole thing is the heart. That's what I love. So obviously this organ is really important. It's involved in pumping blood all around your body. So when the oxygenated blood comes to the heart, and then obviously the blood carries oxygen and nutrients for all our cells. And if we don't have oxygen and nutrients, we can't survive. So without the heart's not beating, we're not living. And the heart beats maybe 80 times every minute. So if you then scale that up to your whole lifetime, the heart beats over 3 billion times in a lifetime, which is an awful lot. So what I specifically look at is can we get the heart to heal itself after a heart attack. So these are the sort of images you might see after a heart attack, someone clutching their chest, really in pain. So what actually happens in a heart attack is the vessels which feed our heart with the blood, so that our heart muscles are getting the oxygen nutrients they need to contract. Those vessels, as we live, and they start to get blocked by these fatty acids that build up in our vessels, and these fatty plaques really form through a variety of factors. It's due to exercise, the things that you eat, good and bad cholesterol, which no one really knows what that is anymore, and some of genetics. But so when you get a blockage in your artery, the downstream areas of heart muscle, those no longer have oxygen and nutrients, so then those muscle cells die, and then obviously you get a really big area of our heart which can no longer function. So what happens after this? So first of all, those muscle cells have died, they're sending off all these big signals, danger signals, ow, we're dead, we need help quickly. And in come our immune system, which are the little soldiers, and then they send in absolutely loads. And then all these little soldiers come in, and they're like these little troops. They'll come and clear up all that debris, all those dead heart muscle cells, and they'll put down these big thing called collagen. And this is these elastic fibers, and they form a big scar. And at first this is really helpful. If they stop our heart actually rupturing, we need that scar to keep the heart in one piece, because obviously loads of the area of the heart has just died. But then that area of scar can no longer contract. So you've got your heart, and it's trying to contract, it's trying to push blood all around your body. It's got this one big area of it which can't contract at all. So if you think you're trying to push a big block of heavy stone up a hill, and then suddenly one of your arms has been taken away, and you're still trying to push this block really up the hill, trying really hard. But obviously this one arm's getting really tired, and at first your heart can do a few things to try and compensate, has all these other mechanisms. So at first you're still doing okay, but as those compensatory mechanisms carry on after the years and years you're living, because your heart is still beating 80 times a minute, those compensatory mechanisms start to fail, your arm gets really, really tired, you just can't push this block up anymore. So we have some drugs and things which try and help you when you've had a heart attack, and try and stop this process as long as possible and keep your heart functioning. but that's not always the case. So, can we fix this real big problem where our hearts aren't pumping as well as we want them to? And that's where the whole field of cardiac regeneration comes into play. And I'm going to hopefully convince you all today that it's really cool. So obviously in your body you've got like millions and millions and millions of cells. And in every type of cell, every cell has like a pack of instructions, which is your DNA. This big molecule contains all the instructions to produce anything that you ever need. So when we start off as a little all our different cells have all the instructions you need to make any type of cell. And as our cells become more and more specialised, they lose the ability to read this whole book of instructions. So obviously when you've got a cell and it wants to become an eye cell, that cell only needs to be able to read the parts of this book which equal making an eye. Whereas your heart cells, they only need to read the parts of the book that equal making a heart cell. So it's not like we don't lose this information, every cell still has this whole book of instructions. It's kind of like we can't read them anymore. Kind of like almost like you've got areas of things which you just can't read, it's not accessible. It's almost like someone's come in your heart cells and glued together all those other pages of the book, so you can only read what's relevant to that type of cell. So, obviously, normally, what happens in a heart attack is we form this big scar, so we still have all that information of how to make new heart muscle cells in our bodies, but we can't access it anymore. Our cells are like, oh, we want new hearts, but it's all just seeing like, cannot access this file, computer says no, etc. But like other areas of our bodies can heal. So if you cut your skin, you form a wound on your, you get a paper cut, that just doesn't bleed forever. You don't have like this massive scar. It can just form new skin cells. Same with like the liver. If you remove like a third of your liver, it can completely regrow. So what we're trying to do is like, how do these other parts of our body which can naturally heal, how can we make that happen in the heart? Because the heart after a heart attack can replace, you lose like 1 billion of your heart cells and it can replace 1% of those cells. And it's like, how can we get that process from 1% to 100% to replacing all that lost tissue? So how on earth would we go about even working out how to do that? So first of all, we look at different animal models that have the ability to regenerate their hearts. So one of them is this little fish, where if you cut away part of its heart, or you give a heart injury, which is this thing called cryo-injury, where we get a copper probe and you dip it in liquid nitrogen so it's like super cold, and then if you put that on the heart, that freezes part of the heart and it's kind of like recreates what a heart attack would be. It'll completely regenerate. So you can cut away part, you can kill part of it with a cryo probe, and it'll completely regrow. The same thing happens in a neonatal mouse, which is just a mouse that has been, once it's just been born, so in its first 7 days of life, if you do the same thing to a neonatal mouse, like either tie off one of its arteries using like a piece of string, or if you use like a cryo injury, where you freeze part of the heart, again it will completely regrow, and you'll have no scar and normal heart. Obviously this is super cool because a mouse is a mammal, so it probably means the same thing is happening in humans, but obviously you'll never know that. No one's going to do that on a baby, that would be crazy. But if you do the same thing in an adult mouse, it will form a big scar like the human. So these models are really good, but what you want to do to work out what's important for regeneration is you want to compare something that can regenerate to something that can't. So for the zebrafish, which can regenerate its heart, we would compare it to like another type of fish, which is sort of similar but can't regenerate its heart. That's not a very good comparison because you're comparing like two different species. It's like comparing like a cat and a dog and trying to work out how they're the same. And for the neonatal mouse, we then compare it to the adult, but again that's really hard because they're completely different developmental time points. It's like comparing the baby to an adult and being like, why don't you work the same? It's just really hard to work out what's going on. So these are the fish that we work on, which I'm going to tell you is like the best model for regeneration that my supervisor found. And they live in these caves in the northeast of Mexico. So they come in two different populations. There's the surface fish and the pachon, which is just a cave in Mexico, and they're called cave fish. And so about 2 million years ago, all these fish used to live in the rivers and streams around these pachon caves. And then it rained a lot one year, and some of the surface fish, as the river levels rose, swam into the caves, and then as the river levels fell again, got trapped in the caves. And since those 2 million years ago, they've diverged into two separate populations. The cool thing about these fish is they're still the same species, so if we want to compare the same two, they have the same genetic material, apart from like a mutations that have acquired over the years, and they're also the same developmental time point, so they're like so much better than the other models. So the surface fish can regenerate its heart. So these are some like heart sections. So this orange, this is all the like heart muscle cells, and this blue, this is that collagen scar I was talking about. So if you give these fish an injury and look at it 21 days after injury, they both form this big scar, which is completely normal and is what we expect. But then again, if you look at it again about 2 months later, the surface fish, all that scar's gone, and you've got like nice, healthy heart tissue, and you wouldn't have it, and it's like it's not had a heart attack. It's like it's not lost any of its heart tissue. Whereas if you look in the Kacham, they can't regenerate their hearts. Something happened in this evolution of 2 million years, which means that they can no longer regenerate. So if you look at them at 2 months or a year later or 100 days later, they still have a big scar like the human. This is a really good model for regeneration, and is what my supervisor has based her whole lab on. So now we're talking about how can we ever use them. So because they're the same species, we can breed them together and they can produce offspring. And you get these weird fish which look like neither of the two surface fish or cavefish. That's because half of these fish come from the surface fish and half of their genes come from the cavefish. So they're like this brand new population. And then if you breed this generation together, so like two brothers and sisters, you get this new generation of fish which kind of look a mix in between both the surface fish and the cavefish. So like the surface fish has like all this pigment like you have in here, but the cavefish, as the years went on, they lost the ability, they don't have any eyes because the caves are black, so they don't need any eyes, there's pitch black, so they don't need them. So you get these fish where they've got some pigment like the surface fish, but they don't have eyes anymore. So you have like a whole mixture of the two because their genomes are a mix of these two original fish. So if you think of, so initially, if you think of regeneration, like the surface fish, they can regenerate, cavefish they can't. So now in this fish, this becomes like a spectrum. So like sexuality is a spectrum. They now have a spectrum ability to regenerate their heart. So you get some who can completely regenerate, some that can't regenerate at all, and some that fall all the way in between. And doing some fancy gene sequencing and statistical analysis, my supervisors identified this whole range of genes and pathways which look like they're really important in regeneration. And that's what I work on specifically, is I work on the immune system and how it's different between the cavefish and the surface fish, and trying to work out how those differences might affect ability to regenerate. And that was it. Thanks for listening.